We had talked about the excretory system, we had talked about the kidney, we have talked about the various steps in urine formation. Now we are going to talk about a mechanism known as countercurrent mechanism. Before we start with the video that is going to be played over here and through which you will understand about the countercurrent mechanism, I need you to keep in mind that over here a reference would be made in terms of osmolarity, and the unit that we would use would be milliosmoles. Alright. Milliosmoles that we are going to use, milliosmole per liter would be the unit that we would be using. Now, if you don't have any idea about osmolarity, by now you haven't read about solutions in your chemistry's knowledge, you just have to keep in mind that more would be the osmolarity, more would be the concentrated solution. Please remember that. If I am saying that some solution is having the osmolarity of 200 milliosmole and other one is having 1200 milliosmole, so you have to keep in mind that the one which is having 200 is lesser concentrated as compared to one which is having 1200, whatever reference I used. The lower the value of osmolarity, it will be lesser concentrated. Higher would be osmolarity, it would be higher concentration of the solution. That means more solutes would be present. I hope I am clear with that. Next thing what we have to deal with is that the countercurrent mechanism, when we study, we talk about loop of Henle like this, which is the tubule itself, the same tubule that we study. The width varies though, the diameter, or we can say that the area of the tubule varies. At some part it is thicker, at some part it is thinner, kidneys part, and it is surrounded by a capillary network that is known as vasa recta. So whatever reference is being made, you have to keep in mind that along with this there is an opposite flow of blood as well. Okay, just for your reference purpose I am telling you that supposedly this is the way in which the urine is passing, the blood would be flowing in opposite direction, and this blood would take up whatever osmolarity changes would be taking place along with the interstitium. This is the interstitial tissue which is present between the loop, and there is a blood supply that is going counter to the flow of the urine that is being concentrated. Now, this countercurrent mechanism plays an important role in concentration of urine. Let us see how it takes place. Now, we are going to see, what you see over here is a kidney. This is what a kidney looks like, very different from what I tried to show you in my own diagrams, very clear over here. We enter the cortex, and over here we see these renal pyramids. Over here we see in the cortex that there is glomerulus, a Bowman's capsule. This is the part where the concentration of urine would take place. This is the capillary network, and vasa recta would be this. Now, this is the formation of filtrate which is taking place. This is the part of Bowman's capsule from where the RBCs are going, and you see that the filtrate is taking up maximum of the ions and maximum of the electrolytes. They are passing into the filtrate through the podocytes. This filtrate is this ultrafiltrate is what we call as GFR. Now, we have to enter into this part, that is loop of Henle, for the countercurrent mechanism. Now, this is the inner part of the tubule, and what you see over here is that there is continuous countercurrent maintenance that is taking place. There is a countercurrent, that one time the fluid is flowing like this, and in the other part the direction is opposite. This is the descending limb of loop of Henle, this is the ascending limb of loop of Henle. That is what, this is the descending, this is the ascending. When we have to understand the countercurrent mechanism, we have to keep in mind that at one stage this flow is totally stopped. We are going to come to that, but understand the kidney's position. As I told you before, the upper part is in the cortex while the lower one is in the medulla. Now, you see the difference in diameter as well. Over here it is thinner, over here it is thicker. Now, what happens is, consider this in a stage where everything is stagnant, that is, it is not moving, the counter current that is flowing is not flowing, it is steady. In that stage, supposedly we are going to start with that. The osmolarity that we are considering, it is 300 milliosmole. This is the thin segment, this is the thick segment. Now, this thick segment has a permeability which takes up the sodium ions which come out, sodium and chloride ions. This thick segment is responsible for taking up the sodium ions. When the sodium ions are taken up into the interstitium, the osmolarity over here decreases, while over here it increases. Please pay attention, the osmolarity has changed. Now, in order to maintain the balance, what happens? These sodium ions, they move inside according to their gradient, and the water moves out. Now please pay attention, because we started from a condition where it is stable, the sodium ions are being absorbed, the osmolarity changes, in return the osmolarity balance is maintained over here. As a result, the water moves out. Now, what is the entire purpose of this mechanism is to restore the osmolarity which was present over here. The same osmolarity should be present over here, because the parts of the kidney, they need to have the same osmolarity. It cannot vary if some sort of fluid is moving through it, and the fluid we are referring is the ultrafiltrate that has passed out of the glomerulus. Now see what we have learnt till now, that some part was getting absorbed from here, then this part absorbed got transferred over here, and over here this osmolarity we are going to see. As it would be absorbed back, this would get less osmolarity. This absorption would increase the osmolarity because it is reabsorbed, and because the counter current is continuously working, it would lead to the concentration and it would lead to the osmolarities as mentioned. This continuous counter current maintenance is responsible for take up of water from here and take up of sodium from here. Whatever water is being taken over here, it is in continuous with vasa recta, this vasa recta which is present. Now this is the descending limb, the water is getting reabsorbed, more and more of water is getting reabsorbed, the osmolarity is increasing. Okay, at the same time, if we consider it in terms of collecting duct is under the control of ADH, which tells if the water is to be reabsorbed more or if it is not to be reabsorbed. So the ultimate concentration of urine also takes place in the collecting duct, which is also in accordance with the osmolarity as we had discussed in the other parts of the kidney. This is the reference of collecting duct. You see more and more water is being reabsorbed. If the ADH says that the water is not to be reabsorbed, then the reabsorption would start and stop, and we'll have more diluted urine. So this is all about counter current mechanism. In this video, what I have tried to illustrate is that the water gets reabsorbed in the descending limb of loop of Henle. As more and more water gets reabsorbed, it is to be counterbalanced by intake of sodium which are absorbed from this zone. Now see this part reabsorbs sodium into the interstitium. As a result, more water is reabsorbed. So somehow the concentration of urine is taking place because water is being reabsorbed. Then because more sodium is being taken up, there is osmolarity restoration because the sodium has been taken up. Though there was loss of water from here, the lot of water was reduced and the osmolarity became higher. Whatever water loss was done, it is restored by take up of sodiums. Okay, and the loss of sodium from here has restored the osmolarity as in accordance with whatever we studied. As we said that over here it was 300, over here it was 300, it was around 1200 milliosmol over here. So the osmolarity is being reduced, and then this counter current mechanism, it enters the collecting duct, and from there the more reabsorption takes place. Now this counter current has capillary supply where the blood concentration is opposite. Whatever water and whatever ions are being reabsorbed, they are kept in accordance with the blood supply. Supposedly blood over here had maximum least water, because you know it all started from glomerulus. In glomerulus maximum water was lost. Now this blood supply takes up the water from here. Whatever water is being lost, it is being given to the blood back in the same coin as it was taken. The water that was taken up, it is given back. Only thing you have to keep in mind that when you start understanding the counter current mechanism, you have to start it from a condition where this flow is stopped, and it starts from here where the thicker segment, it takes up the sodium ions. When it takes up the sodium ion, it goes to the interstitium. The interstitium passes it to the descending limb again. The descending limb then throws out water, and then this counter current goes on and on and on, and the blood which had lost so much of water, it takes up the water. The interstitium which had lost so much of sodium and it could not take up the chloride ions, they are taken back into the interstitium, and because this is going on and on and on, there is concentration of urine which takes place as a result of counter current mechanism. So this is all about the counter current mechanism. I hope it is a bit clear. In case it is not clear to you, like it is little bit confusing, you can just keep in mind that blood had lost its water in the glomerulus. It has to take up its water in any case, and that water would be given back in the loop of Henle. From where? From the descending limb. In order to make the filtrate lose water, the filtrate has to take some ions in it so that the osmolarity changes. Those ions would be taken up when the filtrate is going upwards in the ascending limb of loop of Henle, and that too is maintained by the osmolarity gradient inside the nephron.